I'm going to describe the work that we've been doing, tell you a little bit about NK cells, how I became interested in them, and hopefully convince you that this line of study merits further investigation. So what are NK cells? I guess the best way to describe them is that they're the conventional cell type within a growing field of innate lymphoid cells. So over the last 3 or 4 years particularly, there's been just an explosion in the description, characterization, and testing of these ILCs, or innate lymphoid cell subsets, showing that the innate lymphoid space is just as sophisticated as the adaptive space. So there's an innate lymphoid cell for every T cell subset that you've ever heard described. So traditionally what you would think of an NK cell doing is being the effector cell of innate immunity. So they identify, target, and eliminate stressed, virally infected, oncogenically transformed cells through an integration of activating and inhibitory signals through a series of activating inhibitory receptors. So when they encounter a healthy cell, which is not really expressing stressed ligands, but is expressing high levels of MHC class I, which is an inhibitory ligand for inhibitory receptors. They leave that cell alone, but with viral infection, for instance, you get a downregulation of MHC class I, a loss of inhibitory receptor signaling, and lysis. So how did I become interested in NK cells, and why would I ever think that they're involved in the pathogenesis of PAH? So this traces back to my doctoral thesis work with Duncan Stewart when he was still in Toronto, looking at these early endothelial progenitor cells in EPC therapy for PAH. So we now know that EPCs, despite their name, are neither endothelial nor progenitor. They're more of a monocytic cell type that, however, does have some capacity to influence vascular modeling in rat models of pulmonary hypertension. So my work was looking at human cells into an athymic nude rat model and found that these early EPCs were able to prevent elevations in right ventricular systolic pressure when delivered 3 days post-initiation of disease. So at the time, the idea was that these cells were acting through a home engraft repair type mechanism. But when I looked at fluorescently labeled cells, we could find them in the lungs at 15 minutes post-delivery, but by 24 hours, they were completely gone. So this is odd for a cell type that's enacting a response over a 3-week disease model. So one thing this forced us to do was really look at our model. So these athymic nude rats, while they do lack mature T cells, they have a robust natural killer cell population. So the idea was that maybe if you could, in this case, ablate natural killer cells with an anti-SILO-GM1 antiserum, enhance long-term cell retention. We see here at 24 hours, and actually saw human cell retention in the ablated rats for up to 3 weeks post-delivery. You could enhance the therapeutic efficacy of this cell therapy. But instead, what we found was that we completely eliminated any therapeutic benefit in these cells, which was exactly the opposite of what I expected, and really made me look at 2 things. First was the monocytic nature of our endothelial progenitor cell therapy, and the second was a potential role for natural killer cells in the pathogenesis of PAH. And all that reading led me to pregnancy. So during the first trimester of pregnancy, you get a profound remodeling of the spiral arteries in the uterus from these highly muscularized, tightly constricted vessels to completely amuscular, flaccid sinuses that are stripped away of their CD31 positive endothelial layer, which is replaced with invading extravillous trophoblast. And if you look at these images here, it's funny because it's the exact opposite of what you'd observe in a PAH lung, where you get enhanced muscularization and endothelial cell proliferation. So decidual natural killer cells are absolutely critical to this process. They represent 40% of all cells in the decidua, 70% of leukocytes, and probably the most compelling evidence to suggest that these cells are actually important in vascular remodeling comes from Ashley Moffat's group in Cambridge, and she looks at compound genotypes of these killer immunoglobulin-like receptors, or KIRs, combined in the mother, combined with the genotype of HLA genotype, or MHC class I genotype of the fetus, and showing that this combined maternal fetal genotype can actually influence a mother's risk of developing preeclampsia, which is in itself a disease of pathological vascular remodeling. I don't know why that switched, but so this was interesting. We had a curious finding in a rat model, but what relevance did it have to human disease? And at this point, I had moved to work with Nick Morell in Cambridge, and one of the first things we did was analyze a subset of data that he already had. but hadn't looked at natural killer cells in. So just looking at the two major subsets of circulating NK cells in humans, these CD56 bright cells, CD56 dim cells, which are the main cytotoxic effectors in human blood, showing that in the PAH population we get a reduced absolute count of CD56 dim cells when compared to healthy donors. We didn't see any reduction in chronic thromboembolic pulmonary hypertension disease controls, suggesting that there was potentially a drop in NK cell number. When purifying these cells out, and looking at their functional capacity in co-culture with tumor cells, we found indeed there was a reduction in their lytic capacity as well. And we linked this back to reduced expression of a KIR molecule, KIR3DL1, which is interesting because this is an inhibitory molecule. So you'd expect it to be upregulated if you see reduced lytic activity. But actually there's a whole body of literature talking about an educational effect of inhibitory KIRs on NK cells. And actually if you split human healthy donor-derived NK cells into KIR positive, 3DL1 positive and negative cells, you see this ability of the 3DL1 positive cells to more robustly lyse tumor cells, which is completely lost in those PAH NK cells that do express KIR. Actually, funny enough, a number of the donors did not. So going on to look at mechanism of this inhibition, we started focusing on TGFbeta signaling, showing that in NK cells from PAH patients, not only is there increased TGFbeta signaling, we use PI1 gene expression as a surrogate at baseline, but also in response to TGFbeta. Interestingly, if you take these NK cells out of a PAH patient and culture them for 72 hours, we actually saw KIR recovery in these cells, which we could block by adding TGFbeta to the culture media. We didn't see anything like that in any of the healthy donors that we analyzed. And also looking at elevated production of MMP9 from NK cells from PAH patients. So this was all interesting, but again, we have to ask the so what question. So we've seen that there's an impairment of NK cells in patients, but this doesn't mean that they actually play any role in the pathobiology of PAH. And these are the current questions that we're addressing, both the so what question and also do mutations in BMPR2 influence NK cell development and function? So for the remainder of the talk, I'm just going to focus on this first question, which is ongoing work that we've been pursuing over the last couple of years. And in order to do this, it's important to ask, well, what happens if NK cells are absent? What happens in mouse models of disease? So for this, we've looked at genetically modified mouse models lacking NK cells. And this paper is from Hugh Brady, published in 2009, showing the E4-BP4 knockout mouse. So E4-BP4 is a transcription factor that regulates the progression of natural killer precursors to immature natural killer cells. And it's viewed as or at the time when it was published, was viewed as a major improvement over previous models of NK cell deficiency, which were lacking IL-15 or IL-15 receptor alpha. Mostly because these animals had grossly normal numbers of T cells, B cells, but had a reduction to near elimination of circulating NK cells. Now, subsequent studies have shown that these animals do actually exhibit some functional defects in T and B cell populations, as well as deficits in ILC populations as well, but they still represented an improvement over what had been available previously. And so in order to test out these animals, we developed a fairly standard system of cardiopulmonary phenotyping in mice, using closed chest conductance catheterization, first on the right side, then going into the left side to look at end diastolic pressure and end systolic pressure, and then looking at systemic blood pressure in the aortic arch, as well as assessing left ventricular function by echocardiography. And what we found was actually really interesting. So at 3 months, these animals are seemingly normal. But if you age these animals out to 9 months, over half of them develop elevations in right ventricular systolic pressure. So it's potentially that these animals are spontaneously developing PAH with age. However, when we looked at the Fulton index in these animals, we didn't see an elevation. Now, this is disappointing, but it's actually not that uncommon for spontaneous models of PAH. Actually, if you look at in the literature with BMPR2 knockout in the endothelium or BMPR2 R89X mice in our hands, there's an impairment in right ventricular remodeling in some of these spontaneous models, mostly because potentially the disease is much less severe than what you'd observe inside the chamber of a hypoxic mouse experiment. So we first wanted to see if this PH was secondary to left ventricular dysfunction. Just looking at the echocardiography, it suggests no. We actually saw with the advent of disease an increase in ejection fraction in these animals. So they're actually developing a hyperdynamic left ventricle associated with a reduction in their internal dimension at systole, so that they're contracting more vigorously by 9 months. And then looking at the catheterization of the left ventricle actually started showing us something pretty interesting. So we didn't see any elevation in left ventricular and diastolic pressure in the knockout mice. But if we subdivided the mice based on those that did and did not develop pH with age, we found that the animals that didn't develop pH actually exhibited a reduction in systemic blood pressure. And this is in anesthetized mice, so you have to take it with a grain of salt, but at the same time, they're anesthetized to a similar rate if you go by heart rate. So that really got us thinking about what was going on in these mice. Looking at muscularization, I don't know why this is automated. So looking at alveolar alveolar duct arterial muscularization, we see an increase in muscularization in the animals that do develop disease, not in the animals that don't, suggesting that our muscularization is secondary. But that's not the most interesting thing that we saw in the lungs of these mice. We actually saw this heavy perivascular cuffing of the vessels in the lung, which made us start thinking that maybe there was an issue with pulmonary vascular leak in these mice. So this is neat, but like I said before, E4BP4 is a transcription factor. It has countless effects both within the immune system and within mice generally. So why not look at another model of natural killer cell impairment? And for this, started looking at the NCR1 GFP mouse. So this is NCR1, which encodes the activating receptor NKP46, which we had shown previously in humans, is downregulated in the context of PAH. So in this case, exons 5, 6, and 7 are knocked out of the endogenous locus and replaced with GFP, which is nice because you can use the GFP as a means of tracking NK cells in these animals. And what we found was curious. So we found, when we looked at the heterozygous mice, we found that a large subset of these animals actually did develop elevations in right ventricular systolic pressure, like what we saw in the E4BP4 mice. But if we go to the homozygous animals, it was much less so and not significant versus the wild type. Again, we didn't see any changes in Fulton index, but this is a fairly modest model when it comes to elevations in pressure. So this made me first check our blinding, then triple check our genotyping. But as we did more and more of these experiments, it seemed to hold. And some explanation may come from looking at the left side. So again, we saw no elevation in left ventricular and diastolic pressure, suggesting that the pH wasn't secondary to left ventricular failure. But we again see this drop in systemic blood pressure at systole. So it makes us think that there's a spectrum going on in terms of vascular dysfunction, and somewhere along that spectrum, the animals seem to develop pH. Looking at the muscularization in these animals, we again see increased muscularization, but only in the hets, not significant in the GFP mice, and some degree of this cuffing, again, in both the heterozygous and homozygous mice, which we've done just a preliminary quantification, just looking at wet-dry lung ratios in these animals, suggesting that, again, there may be increased perivascular leak. But again, that was, geez, this thing is driving me crazy. That wasn't the most interesting thing about the GFP mice. A lot of them, actually, this really ruined our blinding. A lot of them seem to develop these engorged cervical lymph nodes, which again suggests altered lymphatic fluid handling, potentially vascular leak in these mice, and lymphangiogenesis. So just with the last couple slides, I wanted to focus on how we're trying to dissect the molecular basis of what's going on here. And for this, it's important to go back to pregnancy. So first off, how is this happening with peripheral NK cells? For the most part, the literature in pregnancy suggests that decidual NKs are specialized subset of natural killer cells, and only they can produce the angiogenic factors that are associated with vascular changes in the uterus. But there's a rising appreciation within the field that decidual NKs may actually originate as peripheral cells that are decidualized upon exposure to TGFbeta from the decidua. So there's also recent works looking at non-small cell lung cancer, again suggesting that the cancer and cancer-derived TGFbeta is corrupting or converting NK cells to an angiogenic phenotype. So just some preliminary work looking at whole lung, looking at gene expression of a number of different angiogenic cytokines. What we found was no difference in a lot of the VEGF-A, VEGF-C, but what we did see was a progressive reduction in angiopoietin-2, which was, again, not what we expected. Angiopoietin-2 is reported to be elevated in a lot of cases of PAH, but when we did confirm it in proteins, suggesting that there was actually a progressive reduction. And what we're looking at now is having to track that back to see if it's a primary effect of NK cell dysfunction in these animals or if it's a secondary effect in the lung, and how this impacts on pulmonary vascular integrity. So just to summarize, first off, just to state that NK cells are known to influence vascular remodeling, particularly in pregnancy. We've previously shown that PAH patients exhibit reduced NK cell numbers and impaired function linked to this KIR3DL1. And here I've shown you that we've observed spontaneous development of PH in 2 independent genetically modified mouse models of NK cell deficiency, and both models also developed or demonstrated a reduced systemic blood pressure in a subset of mice. So I'd like to thank Nick Morrell, particularly at Cambridge, and my new colleagues, including Stephen Archer at my new home at Queen's University in Kingston, Canada. Thank you.
That was great work. Can you integrate your understanding of NK biology with the traditional understanding of NKs recognizing self class 1 and how that might be the traditional understanding of how NKs work, how that might integrate into PAH?
Oh, well, it's difficult. Well, I guess if you look at I again go back to pregnancy and look at the types of HLA molecules that are expressed on trophoblast, HLA-C, HLA-G, and the idea that these MHC class 1 molecules are somehow switching off certain elements of NK cell programming, NK cell cytotoxicity. There was a great paper in PNAS that came out just recently looking at HLA-G expression in the decidua in trophoblast cells. I'd love to look at HLA-G in the lungs of PAH patients. Somebody do that, please. And the ability to kind of convert their programming from a cytotoxic state to a vascular remodeling agent. So that's where pregnancy is at now. It's quite frustrating, actually, because the role of NK cells in pregnancy has been appreciated for 20 years, and they're still no closer to really understanding. First off, half the field doesn't believe it. The other half believes it very strongly. And there's still very little appreciation of how, mechanistically, how that's all happening. So I'm trying to follow their lead and work it out.
Do you know if it's possible to integrate or to evaluate class 1 molecules on explanted tissues in the way that they do with,
You have to be very careful about the antibodies that you use, and in terms of which antibodies recognize which classical and non-classical class 1 molecules, particularly HLA-C, which is thought to be restricted to trophoblasts. Some have reported that it's expressed in certain cancers and potentially on endothelial cells in some tissues, but I think that relates back to poor use of non-specific antibodies. No, it's definitely important to subdivide class 1 molecules. We've done that a little bit on BOX from PAH patients, looking at actually elevated levels of HLA-B, which is the ligand for KIR3DL1 on BOX from PAH patients. Haven't published that yet.